want to talk about something that's known as deoxyribose nucleic acid. Of course, you probably remember this from high school biology. So deoxyribose nucleic acid, in other words, in short, we call it DNA. All right, and this is simply just the genetic material, all right, or the information that all of our cells have, the information that all of our cells have to make things, to make different types of enzymes, proteins. In DNA has instructions to make our nose, our mouth, our teeth, our stomach, the different enzymes that our liver uses, the different enzymes that's involved in the digestive process, the different enzymes and proteins or anything that bacteria makes, trees, dolphins. All living organisms have DNA. All right, so in terms of eukaryotes are organisms that is termed of having a true nucleus. So here's a nucleus, all right? And the nucleus is just actually the compartment that stores DNA, all right? So that's in eukaryotes. So the DNA is stored in the nucleus. In prokaryotes, that's bacteria. Bacteria does not have a nucleus, but of course they still have DNA. So it's just in the cytoplasm of the bacteria. So all organisms on the face of the earth are eukaryotes. Every organism on the face of the earth are eukaryotes, except for bacteria. Bacteria are prokaryotes, but beyond the prokaryotes, everything else, fungus, yeast, mold, mushroom, plants, animals, protists like algae, euglena, paramecium, all those guys are eukaryotes. All right, so again, DNA, we have a cell, okay? So if you kind of go like from big to small, we have a cell. And then in the cell, we have chromosomes, okay? And then if you break the chromosomes down further and further down, and then it gets down to the DNA. So the DNA is actually all wrapped up and intertwined in the chromosomes, and we have a grand total of 46 chromosomes in every cell of our body, except for our gametes or sex cells, and I might get into that when I talk about meiosis. Again, chromosomes, and then if you really kind of get into the nitty-gritty in that regard, boom, DNA. All right, these are the two scientists actually who discovered the double helix structure. So DNA is actually, structurally speaking, it's kind of like a winding staircase. These two scientists, Watson and Crick, and these were British scientists. Actually, one was the scientist, and the other one was the mentee, the graduate student. So Watson and Crick. All right, so this is kind of like that winding staircase which I talked about, DNA molecule. All right, so actually that's what it actually looks like. So you have the backbone part, that's kind of like the light blue part, and then the bases that you see here, all right, kind of like the rungs of the ladder. And in the bases you have four bases: adenine, thymine, guanine, and cytosine. Adenine binds with thymine, guanine binds with cytosine. So again, here's the DNA structure again. All right, this is just kind of an unwound staircase, but then here it just has the staircase. And again, as you see here, this thing keeps on popping up here, the images. So when you see this, that's just where I got the image. Nonetheless, adenine binds with thymine, guanine binds with cytosine. So these are what we call complementary bases. So the guanine binds with cytosine, adenine binds with thymine. So the complementary base to guanine is cytosine. The complementary base to adenine is thymine. Here it is again. So again, cytosine and guanine. So you cytosine and that's, hey, what's the complementary base to cytosine? Guanine. What's the complementary base to thymine? Adenine. Okay. Are there cases where thymine will bind to a guanine? Could that happen? Yeah, it's possible. That gets into like the DNA mutations and all that stuff, which of course happens. but typically speaking, generally speaking, these are just what we call the complementary bases: thymine to adenine, guanine to cytosine. And this thing right here we call the backbone chain. The backbone chain here, okay? And that's the sugar molecule and the phosphate. All right, so the deoxyribose sugar, phosphate, deoxyribose sugar, phosphate, deoxyribose sugar, phosphate, and again, these are the bases that are in the rung of the ladder. So again, phosphate, the sugar, deoxyribose sugar, and then the base, and that base again can be either, switch this here, adenine, or it can be thymine, or it can be cytosine, or it can be guanine. All right, this is just kind of for those of you chemists out there, if you just kind of want to look at the chemistry of it there, right? This kind of gets more in depth of the chemistry of it, the nitrogen, the oxygen, the double bonds here between the nitrogen and the carbon here, but we don't have to get that deep into that. Okay, so this is something we call the central dogma. So as it relates to DNA, DNA has the opportunity to make a copy of itself. We call that DNA replication or DNA synthesis. So we simply just make a photocopy of itself. Or if it doesn't, DNA actually will be used to undergo what's called transcription, or in other words, RNA synthesis. So that's where it makes RNA, messenger RNA. And then DNA, and all this is going on in your cells. Okay, this part is going on in the nucleus of our cells. And then once RNA is made, and then translation takes over. So RNA is used to make proteins, and this is the end product. Okay, so again, this lecture here, I'm going to get a little more into the whole central dogma process. Well, I'm not going to get into the whole process or anything. I'm just going to kind of give you like a real kind of surface understanding and idea of, again, how this works. And with some of you guys, it would be like a nice reminder. Some of you might say, "Oh yeah, that's right, I remember that." And some of you guys, maybe it might be some new information that I present. So again, look at DNA. We call DNA the blueprint of life. So just like blueprints, okay, has all the information like, hey, here's a blueprint of such and such. Now we need to build it. But the blueprints has all the specifications in terms of how something's built. Like the blueprint of the Batmobile. I just saw that. So, let me just toss this in here. Actually kind of cool. So blueprint of a Batmobile, or something that's more realistic. All right, this is supposed to be like a blueprint of a house. All the dimensions, what needs to be used in terms of, like, you know, the plumbing system throughout the house, HVAC, all that stuff. All right, so here's the blueprints of the house. Something like DNA. So think of DNA as this. DNA has the information or the recipe. So just like you have a recipe, let's say for bran muffin, DNA says, okay, here, you need to add this, you need to do 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 this, and you need to do this. So it has all the directions, all right, of how to do things. Okay, so that's the purpose of DNA. The final product is the protein. That's the final product, blueberry bran muffin. Okay, so of course, you know, so you have like the DNA here, and then you go undergo transcription, or in other words, RNA synthesis, and then translation or protein synthesis for the final product. All right, so to kind of make it simple here, DNA, the blueprint. This step right here is a step of the building materials and the workers. Okay, and then here the translation or protein synthesis is the end product, the house, the end product. This is just an example of DNA sequence here. So you have like adenine, adenine, guanine, cytosine, thymine, thymine, adenine, thymine, cytosine, adenine, adenine, guanine, adenine, 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 so on and so forth, right? So here it's not showing the complementary bases. But if I were to ask you, hey, well, you know, what would be the so if I said, like, hey, what would be the complementary base of adenine? It would be You know, I say like thymine, complementary base of cytosine. I'm sorry, guanine, cytosine. Complementary base of cytosine, guanine, adenine, so on and so forth. But it's just not showing. It's just only showing one side. It's not showing the complementary base or anything. It's just showing one side. But of course, it's just real easy to do the complementary bases. Okay, so this is kind of quite interesting. What percentage of the world's population, despite region, culture, ethnicity, etc., are genetically identical? So we have, of course, just cultural diversity here on planet Earth. So let's see here. So again, what percentage of the world's population, despite region, culture, ethnicity, are genetically identical? So think about it. What do you think? So think about what percentage, if you just randomly selected someone from, I don't know, from one part of the world who's totally is totally different. Well, I say totally different, but ethnicity, they're different from another person from another part of the world. Let's say someone from, I don't know, Colombia compared to someone in Japan, or someone in Kenya compared to someone in who lives in the Balkans, or someone who actually lives in Australia comparing that to someone who lives in Mexico. What percent difference would you say that we are here on planet Earth? So here's cultural diversity here, a reflection of the globe. You thought about it? Well, guess what? We are 99.9% genetically identical. I've even read a report where that they says it can be upwards to 99.99, but 99.9. And this came from the National Human Genome Research Institute. Okay, that's high, and I'm sure that that's. Oh, and of course, you know this sign like greater than or equal. Okay, and I'm sure it probably surprised a lot of you. I mean, I pose this question to students, you know, and they might go as, okay, well, we're 5% genetically identical, or okay, 50%, 75%. No, we're 99.9%. And of course, that might amaze some people. Why? Because basically, that very small percentage of our differences is that what you see on the outside. Think about it. All human beings. We all have kidneys, livers, tibia, fibula, radius ulna, carpals, metacarpals, like in our hands. We're all producing the same digestive enzymes. I mean, you got to take all that into account, where that it's actually basically genetically similar, the same. In fact, I mean, add this. It's been interesting. My cousin, he's an African American male, and he received 2 kidney transplants in 2 different time periods. His immediate family was not a close enough genetic match for him to receive one kidney from one of his family members, his parents or his siblings. Some of his cousins even got tested, you know, or other cousins and everything, were not a close enough genetic match. My cousin, again, an African American male, received 2 kidneys at 2 different times from 2 Caucasian women. These 2 Caucasian women were a closer genetic match to my cousin than his own intermediate family members, biological family members there, right? So I think it's very interesting. I think it's very fascinating. And you guys, and that is not uncommon at all right, in terms of transplant medicine. That's something that's not uncommon at all. So it just, again, goes to the fact that it's showing that, wow, basically how close genetically, okay, despite maybe, you know, the differences in terms of race, culture, ethnicity, and so on and so forth, how close we are genetically speaking. It's amazing. All right, thank you.